I'm really excited to be here at the CRISPRCon workshop. I really enjoyed all the presentations and discussions thus far. And today I'm going to talk to you about how we can use CRISPR to prepare for future mosquito-borne pandemics. So I wanted to start off with this slide. This is a slide from Gates Notes. It just emphasizes that mosquitoes are essentially the deadliest animals on Earth. They kill more people on Earth than any other animal combined. The next closest thing is humans killing each other because we love to go to war. So why are they so deadly? Well, they're so deadly because they transmit so many different pathogens that can affect human health. On the left here, we have malaria. It's estimated that there's about 1,000 deaths per day from malaria. And these are unfortunately mostly children under the age of 5. This equates to about a child dying every 2 minutes, even today. We also have dengue fever that's transmitted primarily by Aedes mosquitoes, Aedes aegypti and Aedes albopictus on the bottom here. It's estimated.
Sorry, Omar, I just muted you. It was an accident. I was trying to let Matt, our third speaker, in. So I think you have to unmute yourself. There you go. Apologies, okay. Everybody.
You can hear me now, right?
Yeah, you're back.
Okay, cool. Yeah, dengue fever, which we have about 390 million cases per year. We have West Nile virus, which is transmitted here in the United States, about 30,000 cases per year. Yellow fever, about 207,000 cases per year. We also have Zika virus. It's estimated that half of the world is at risk of being infected by a mosquito-borne pathogen at the moment. So I did this interesting interview earlier this year with David Lazarus, and he wrote this column in the LA Times titled, "COVID isn't spread by mosquitoes, but the next pandemic might be." We really dodged a bullet here with COVID not being transmitted by mosquitoes. I mean, COVID is already a horrible thing we're all facing, but imagine if it were also transmitted by mosquitoes. It would just be that much worse. So we got really lucky, but maybe we won't get so lucky next time. And the last epidemic that we had was in 2015, 2016, mainly in South America, where we saw the Zika epidemic. And we had pictures like this all over the news where babies were being infected with Zika virus and getting microcephaly. This was a very scary time. And fortunately, we're not facing Zika at the moment. But again, we don't know what's going to happen in the future. So we really need to prepare for future epidemics, pandemics, breakouts, etc. I wanted to point out that climate change is actually affecting the increased habitable range of mosquitoes. So on the left here, you can see a diagram of California, and you can see these yellow dots that are popping up. This is the spread of Aedes aegypti, which is a major dengue vector throughout California. And it was introduced in 2013 via cargo ship. And now it's basically spread throughout the entire state. And it's causing a lot of problems because it's a really nasty mosquito that bites during the day and also transmits diseases. I see it in my backyard here in San Diego. It's very hard to manage because they breed in very small containers. But this was recently introduced and spread throughout the state. And on the right, you can see the increased risk of disease transmission. The current risk is on the top of Aedes aegypti, Aedes albopictus. And if you see in 2050 and 2080, this risk is just increasing. And that's due to climate change, warming up the environment, creating a more habitable environment for these mosquitoes to breed and rear in. So the question is, how can CRISPR be used to protect us from mosquitoes? And there's actually a lot of things that are being done in this space. I wanted to focus your attention on something that our lab is developing and we're really excited about. And we think this is a technology that can be potentially seen in the field relatively soon. And it uses CRISPR. So let me first describe how this works. So it's based on this premise of using this old technology called sterile insect technique, where what one does is they sort of mass rear their insect of choice. They separate the males from the females and they sterilize the males. They release the males into the environment, and those sterile males will mate with wild females that are in the environment. And that will result in no progeny being produced. And this can result in population suppression and elimination. It's been very successfully used on a number of agricultural crop pests. On the bottom, you can see a couple examples here: the screwworm, the Mexican fruit fly, the medfly. Some of the limitations of this approach is that the irradiation, it induces a fitness cost to the males. So it makes them, for some insects, this is actually makes this technique not effective. It doesn't work well for mosquitoes, and you must release adults, which makes it a little bit difficult to scale for some species.
5 minutes.
Great. So on this here, what I'm showing you is that existing technologies which rely on SIT essentially use, you have to mass-free your insect in a factory, and then manually you release the adult males that are irradiated into populations for suppression. This makes it difficult to scale because you have to actually build these factories all over the place because you can't really move these males around very far. They're very fragile. So what we've been thinking of is how we can actually design a system that uses CRISPR that can enable the development of one factory, and which would allow you to release eggs into the environment using drones, which would increase scalability of the system. And so we came up with this system here, which is extremely simple and effective. And on the top here, I can explain how this works. So we essentially genetically encode the CRISPR components into the genome of the mosquito. And we have two different lines we generate. We have a guide RNA expressing line and a Cas9 expressing line. And the guide RNA expressing line encodes guide RNAs that target genes important for female viability and male fertility. And the Cas9 expressing line just expresses Cas9. So separately, these don't really do much because the Cas9 and guide RNA need to come together in order for the system to work. So you can maintain these lines separately in a facility and then simply use robotics to cross them together. And then the result of that is the progeny essentially will only be sterile males. And you could deploy these into the environment at the egg stage, which is really great for a mosquito like Aedes aegypti, which the eggs actually go through a diapause. So you can store them up to a year and then simply drop them in water and they hatch out as a sterile male. So this system has a lot of really great benefits in that it's logistically scalable. You can design a factory to mass produce these eggs and then deploy them into the environment. It's environmentally friendly. It doesn't rely on insecticides. It's evolutionarily stable. The components are separated from each other in a factory, then crossed together, and the result is a dead end. It's a sterile male. It's safe. It's not predicted to persist or spread in the environment. It's regulatable. And I put that word in because a lot of discussion using CRISPR for insect control is talking about gene drives, which at this moment is unclear whether one can regulate a technology that they can't control. It's effective, also controllable, because again, it's just a sterile male. It's there for one generation, then it falls out. Species specific, you're only targeting a single species, unlike an insecticide where you're gonna spray and kill beneficial species like honeybees and things like that. And I wanted to point out this is a platform technology. We've designed this in mosquitoes, also in fruit flies. So it can be used for not only human disease, but also for agricultural crop pests. So, going forward, this is kind of my final slide, but I wanted to show like the innovation that's happened in electronics. So on the top, you see the cell phone of the 1960s. I didn't have one of these, but maybe some of you may have. That's what it looked like, but you see that the cell phone of today, this is the Apple iPhone 13. There's a lot of innovation there. On the bottom here, you can see how mosquitoes were controlled in the 1960s and even prior to that. And on the right here, this is actually a image from a news channel in Florida during the Zika epidemic, where they were doing aerial spraying of insecticides over houses. So there's not much innovation here in terms of how mosquitoes have been controlled over the last 50 years. And so I think going forward, we're going to see more of this, using insects to control themselves. And CRISPR has really empowered this development of many different approaches. And just the one I showed you today is just one example of how this can be done. That said, I don't have much time left. I just want to thank you for your time. Obviously, thank my lab and all of our funders.